So as we move into the afternoon, our first speaker is Crystal Prom. She grew up in Franklin County, Virginia, completed her bachelor's at Virginia Tech, double majoring in animal and poultry science and dairy science. When she was at Virginia Tech, she worked as an undergrad researcher in Dr. Ben Corral's lab, conducting various lipid-related research. And when she graduated, she moved to southeastern Wisconsin and worked as inside sales and sales administrator for Caftel for 2.5 years before she went back to school. She received her master's at University of Illinois with Dr. Drakely before she moved to Michigan State University to work with Dr. Adam Locke for her PhD. And I want to wish her a special congrats because I understand just a little over a week ago, she successfully defended that PhD. And she'll be moving back to southeastern Wisconsin to work for Cargill as a dairy nutrition consultant pretty soon. So with that, Crystal, the floor is yours.
Thank you very much for that. You can hear me okay? Okay, great. All right, I know we're running a little tight on time, so we're going to get going here. This that I'm presenting today is a little bit of research that was from my dissertation. So my dissertation work focused on the effect of oleic acid and exogenous emulsifiers on fatty acid absorption in lactating dairy cows. I want to begin today by talking about why fatty acid absorption is important in our dairy cows, as well as discuss a little bit of the physiology of fatty acid digestibility and absorption in ruminant animals. And then we'll explain what exactly emulsifiers are before we go into our two potential strategies for improvement that I investigated. The first being whether oleic acid can improve the digestibility of other fatty acids, and the second being whether supplemental exogenous emulsifiers can impact the fatty acid digestibility. And then I'll finish with some practical considerations. Basal diet of dairy cows are naturally low in fatty acids because the feedstuffs contained in the basal diets are low in fatty acids. However, as our modern dairy cows continue to increase in milk yield, they're also often increasing their dry matter intake. So overall, their total fatty acid intake is increasing. Additionally, we are often supplying supplemental fat in the dairy cow diets in order to increase the energy density of the ration to support the yield of milk and milk components, as well as other energy requirement needs. So between increasing the dry matter intake and supplying additional fat, we overall are increasing the fatty acid intake of these cows and thus the amount reaching the small intestine to be absorbed. However, some of the work that Dr. Borman did during her PhD was a meta-analysis, and she found that as we're increasing the amount of fatty acids reaching the duodenum of the small intestine, we actually decrease total fatty acid digestibility. So we're providing these fatty acids to the cow because we want them to be able to absorb them and use them for energy. However, the more we provide, the less they're actually absorbing. So we want to investigate and understand how these are impacted, how this digestibility is impacted, and how we can improve it. Backing up a little bit, I want to go over how exactly the digestibility and absorption of fatty acids occurs in the ruminant animal. So the fatty acids consumed by the cow are primarily linoleic and linolenic acids in the form of triglycerides or glycolipids. If any fat supplements are included, they will be either free fatty acids or triglycerides. The microbes in the rumen will begin the process of digestion of fatty acids with hydrolysis and biohydrogenation. So although the linoleic and linolenic are the primary fatty acids coming into the rumen, the fatty acids leaving the rumen are primarily the saturated stearic acid in free fatty acid form. There will also be microbial phospholipids leaving the rumen, as well as any inert fats. These fatty acids will flow to the small intestine and be absorbed onto feed particles at that time as they're coming from the rumen. However, in order to be absorbed, they need to pass from that feed particle through the aqueous environment to the surface of the small intestine, where they can then be absorbed by the enterocyte. But because they're hydrophobic, these fatty acids need to be transported across this aqueous environment by the micelle. And these micelles are formed by lysolecithin and bile salts. And the bile salts are coming from the liver, and the lysolecithin is formed from either biliary or microbial lecithin, with a fatty acid then cleaved off by pancreatic phospholipase. So these micelles are important to solubilize those water-insoluble fatty acids to transport them across that aqueous environment. And the long-chain saturated fatty acids will be located in that hydrophobic inner core of the micelles. Emulsifiers, such as the naturally produced lysyl lecithin and biosalts, promote this micelle formation. The absorption process will occur following the uptake of fatty acids by the micelles. The micelles will transport it to the surface of the enterocyte, and then the fatty acids will enter the enterocyte by passive diffusion, but there is research that indicates this could be affected by various transport proteins. Once inside the enterocyte, the fatty acids will be resynthesized into triacylglycerol in the ER, and then will be packaged into either chylomicrons or VLDL, and then enter circulation via the lymphatic system. Previous research has indicated there are differences in the digestibility of different fatty acids. So going back to the meta-analysis that Dr. Borman did, she observed a strong negative relationship between the duodenal flow of stearic acid and the digestibility of stearic acid. And recall that stearic acid is the predominant fatty acid in the small intestine available for absorption. So this negative relationship here has a large overall impact on total fatty acid digestibility. Jackie then followed this up with a feeding study where she fed different amounts of stearic acid and again saw that strong negative relationship. So as we're increasing total fatty acid intake with our stearic acid supplementation in this case, we decrease total fatty acid digestibility. Rico et al. Then performed a very similar study. However, they utilized palmitic acid, and you can see that this relationship is much less pronounced when we are feeding palmitic acid versus stearic acid. So there are differences between the fatty acids on digestibility. Jonas D'Souza then looked at this further, and he fed different ratios of palmitic, stearic, and oleic acids. So in this study, he had the non-fat supplement control, and then he had 3 diets that contained fat supplementation at 1.5% diet dry matter. The first treatment was 80% palmitic. The second was 40% palmitic and 40% stearic. And the third was 45% palmitic and 35% oleic acids. And he observed that the palmitic acid supplemented diet was able to maintain the same total fatty acid digestibility of that of the non-fat supplement control. However, when stearic acid was introduced, it decreased total fatty acid digestibility by 10 percentage points relative to the control diet. Interestingly, the introduction of oleic acid in this study increased total fatty acid digestibility by 2 percentage points compared to that non-fat supplemented control. So again, here we see that there are some quite strong differences between fatty acids on total fatty acid digestibility. Jonas D'Souza then followed this work up with another feeding study that fed different ratios of the palmitic and oleic acids. And he observed that there was a tendency for increasing oleic acid to increase total fatty acid digestibility. So of these 3 fatty acids examined, there are indications that oleic acid has a beneficial impact on digestibility. This is likely due to the potential role of oleic acid as an emulsifier, which will then help with that micelle formation. Dr. Freeman in the 60s and 70s examined a lot of solutes impact as emulsifiers, potential as emulsifiers, and he found that oleic acid is half as amphiphilic as that naturally produced lysyl lecithin. And this means that oleic acid does possess some characteristics that would make it a good emulsifier for increasing micelle formation. He also examined different fatty acids' effect on critical micellar concentration and saturation ratio. CMC is defined as the amount of a substance needed before the micelles will begin to form. So you can see that oleic acid has a much lower CMC than palmitic or stearic acid, meaning that less oleic acid is needed in biosalt solution before those micelles begin to form. He also looked at saturation ratio, which is the amount of a substance that the biosalt can hold. So the biosalt solution can hold much more oleic acid compared to the other 2 fatty acids, palmitic and stearic acid. So we see that oleic acid does have potential as an emulsifier to improve micelle formation. So what exactly is an emulsifier? I just wanted to touch on this a little bit because it's the foundation to a lot of my research. Emulsifiers are substances that stabilize droplet formation. Often they do this by reducing surface tension, in which case they're referred to as a surfactant. They're often amphiphilic, meaning they have a hydrophilic head and a hydrophobic tail. And these emulsifiers allow immiscible liquids to form an emulsion. So here's an example on the right. You can see an example of oil and water. And if you think of a bottle of oil and water, such as salad dressing, if you shake it, these two substances will temporarily disperse in each other, but will quickly separate back out. However, if you add a surfactant, it will stabilize the interface between the two substances and it'll allow a stable emulsion. So they will not separate back out. They will stay dispersed within each other. Interestingly, in my research, I found that the term emulsion actually comes from the Latin to milk out, because milk is a key example of an emulsion, with the milk fat droplets suspended in water, and the milk proteins actually act as emulsifiers. So with my work, I had two main questions that I aimed to answer. The first being, can oleic acid improve digestibility of other fatty acids? And the second being, if more fatty acids are flowing to the small intestine, does emulsification become a limiting step to fatty acid digestion? And then can supplemental exogenous emulsifiers improve fatty acid digestibility? I'm going to first focus on the first question regarding oleic acid. So at this point, Alicia Birch had not performed her study yet. So to the best of our knowledge, no one had directly compared stearic acid and oleic acid in supplemental fat blends. They are both 18 carbon fatty acids. So this is something to consider, as oleic acid can be biohydrogenated in the rumen to stearic acid. So we wanted to directly compare different ratios of stearic and oleic acid in supplemental fat blends and examine these effects on fatty acid digestibility and production responses of dairy cows. We hypothesized that increasing oleic acid at the expense of stearic acid will increase total 16 carbon and 18 carbon fatty acid digestibility. In this study, we had 8 cows that were assigned to a replicated 4x4 Latin square design. And the treatments were a control diet with no added fat, and then 3 supplemental fat diets at 1.5% diet dry matter. We had different ratios of stearic and oleic acids. So we had the control treatment, and then 50% stearic, 10% oleic, 40-20, and 30-30. And these blends were achieved by utilizing 4 different fat supplements, and palmitic was held constant at 33% in these blends. We fed a typical Midwestern diet, and our fatty acid supplements were substituted for soy hulls. For our basal diet, the fatty acid content was 1.8%, and the inclusion of fat supplement brought this up to 3.2%. Our periods were 14 days long, with the sampling occurring on the last 4 days. Our sampling periods consist of milk sampling twice a day, feed ingredients and ort sampling daily, feces every 9 hours, blood once, body weight on the last 2 days, and the cows were body condition scored on the last day by 3 trained investigators. Our milk samples were analyzed by DHIA for components, and fatty acid profile was determined in our lab by GLC for milk, feed ingredient, orts, and feces. Total tract digestibility was determined using indigestible NDF as a marker. Moving into results, my graphs will be set up similarly with the treatment on the x-axis and the variable of interest on the y-axis. I will have the p-values of 3 pre-planned contrasts underneath the graph. For this first study, my pre-planned contrast was the control versus the average of the 3 fat treatments, and then the linear and quadratic effect of increasing oleic acid within those fat treatments. You can see that in this study, fat supplementation had no effect on dry matter intake. However, logically, the inclusion of fat increased total fatty acid intake. However, you can note that there was no difference between the 3 fat supplemented diets. Looking at the variable we're most interested in, total tract fatty acid digestibility. Overall, supplemental fat decreased total fatty acid digestibility compared to control. However, within the fat supplements, increasing oleic acid linearly increased total fatty acid digestibility, with it being 12 percentage points higher on that high oleic 30-30 treatment compared to the high stearic 50-10 treatment. We saw similar patterns with our 16 carbon and 18 carbon fatty acid digestibility, with the 30-30 treatment increasing them by 11 and 13 percentage points respectively compared to control. So there's indications here that increasing oleic acid doesn't just improve that 18-carbon fatty acid digestibility. Remember, it's an 18-carbon fatty acid, but it is also improving 16-carbon fatty acid digestibility, likely due to improving micelle formation. Fat supplementation overall increased total absorbed fatty acids because we were providing more fatty acids in the diet. And within the fat supplements, increasing oleic acid linearly increased total absorbed fatty acids. Recall that there was no difference in total fatty acid intake between these three diets. However, the high oleic treatment increased the total absorbed fatty acids by more than 100 grams per day compared to that high steric 50-10 treatment. Looking at our production results, we see that overall supplemental fat increased milk yield by 2.2 kilograms per day compared to control. And overall, supplemental fat also increased milk fat yield by 0.08 kilograms per day compared to control. In summary, for this first study, increasing oleic and decreasing stearic acids in a supplemental fat blend did not affect dry matter intake, but it did increase digestibility and absorption of fatty acids. Increasing oleic decreased milk fat content, but did not affect milk fat yield. Overall supplemental fat increased milk production regardless of fatty acid profile. So in this study, it was unknown exactly how much oleic acid was reaching the small intestine because of potential biohydrogenation in the rumen, but we do know that increasing oleic acid in the diet improved fatty acid digestibility. However, we did wonder if we removed the rumen from the equation and abomasally infused oleic acid, what would the direct effects be on fatty acid digestibility if we knew exactly how much oleic acid was reaching the small intestine? So the objective for our next study was to determine the dose response effect of abomasal infusion of oleic acid on fatty acid digestibility and production responses of lactating dairy cows. And we hypothesized that infusing oleic acid would increase total 16-carbon and 18-carbon fatty acid digestibility, as well as increase the yield of milk and milk fat. In this study, we used 8 cannulated cows that were assigned to a replicated 4x4 Latin square design. We infused oleic acid at doses of 0, 20, 40, and 60 grams per day, and these infusions were broken up into four infusions per day. Again, we've had a typical Midwestern diet, and we did include a saturated fatty acid supplement in order to bring the total dietary fatty acid content up to 3.3%. In this study, we had 18-day periods. The first 7 days was a washout period with no infusions. And then for the next 11 days, we infused our oleic acid every 6 hours. And the sampling period, again, was in the last 4 days. And the samples were analyzed in the same manner as in that first study. In this study, our pre-planned contrasts were the linear and quadratic effect of increasing oleic acid, and then we also had a comparison between the 0 and the 60-gram doses. As with the previous study, there was no effect on dry matter intake, and again, logically, as we increased oleic acid, we increased total fatty acid intake. Looking at total fatty acid digestibility, we saw that oleic acid linearly increased total fatty acid digestibility, and it was 8 percentage points higher on that 60-gram per day dose compared to the 0-gram dose. As with the first study, oleic acid increased both 16-carbon and 18-carbon fatty acid digestibility, with it being 7.7 and 8.2 percentage points respectively higher compared to control. So again, we see that oleic acid, even though it's an 18-carbon fatty acid, it's also improving 16-carbon digestibility. Increasing oleic acid increased total absorbed fatty acids, with it being 145 grams higher on that 60-gram dose. And I wanted to point out that in each case, the amount absorbed was higher than the amount infused. So for example, if we look at a 60-gram dose here, if the cow absorbed all 60 grams that we provided to her, she still absorbed 85 grams per day additional dietary fatty acids. So this oleic acid is improving dietary fatty acid absorption, likely again due to that micelle formation. This translated to a tendency for increasing oleic acid to increase milk yield, and it was milk yield is 3.7 kilograms per day higher on the 60-gram dose compared to the 0-gram dose. There was no effects on milk fat yield in this study. So overall, for the second study, abomasal infusion of oleic acid did not affect dry matter intake, and it did increase fatty acid digestibility and absorption. It tended to linearly increase milk yield and energy-corrected milk and did not affect milk fat yield. So now we've answered that first question. We've seen that oleic acid consistently improves digestibility of other fatty acids. Now moving on to the second one. If there are limitations to the cow's natural capacity to emulsify and form those micelles, can supplemental exogenous emulsifiers improve fatty acid digestibility? So in this third study, I just wanted to remind you that as we're increasing the milk production of our cows, we're often increasing their dry matter intake as well. And we often include supplemental fatty acids in their diet. So between these two, we are increasing fatty acid intake of the cows, which has been shown to be related to a decrease in total fatty acid digestibility. Recall that the cow requires both bile salt and lysolecithin to form these micelles that are necessary to transport fatty acids to the surface of the enterocyte. But at some level, perhaps we are providing more fatty acids than the cow can naturally emulsify into these micelles. So we proposed that providing exogenous emulsifiers could aid the cow in micelle formation and thus fatty acid absorption. Our lab group began to examine the use of polysorbate, known as the trade name tween, which is a non-ionic surfactant emulsifier that's used commonly in pharmaceutical and food industries. And some of you have maybe perhaps even used it in lab settings. It consists of a polyethoxylated sorbitant base esterified with a fatty acid tail. Jonas D'Souza led a study with Moran Western where they abomasally infused various doses of the tween 80, and they observed that at a dose of 30 grams per day, they improved total fatty acid digestibility by 10.2 percentage points. So they did show that this tween 80 has the potential to improve total fatty acid digestibility. However, there are different types of tween available, and they all have the same polysorbital base, but they differ by the fatty acid tail attached. So tween 40, the tail is predominantly palmitic acid. Tween 60, the tail is predominantly stearic acid. And tween 80, which is the one we used in the first abomasal infusion study that I just showed, the tail is predominantly oleic acid. So since we know that different fatty acids affect digestibility, we wanted to know whether these tails attached to the tween would affect fatty acid digestibility. So the objective of our third study was to compare the effect of different tweens on their ability to improve fatty acid digestibility. We hypothesized that all of the tweens will increase fatty acid digestibility relative to control, and that the increase in fatty acid digestibility will be greatest for tween 80 because of that oleic acid tail. Our materials and methods were the same as in the study I just showed, but for this study, the treatments were 30 doses of 30 grams per day of either water or tween 40, which when we processed it in our lab, we found was predominantly palmitic acid. Tween 60, the fatty acid tail was 49% stearic acid and 43% palmitic acid. And with tween 80, the fatty acid attached was predominantly oleic acid. For the results for this study, our three pre-planned contrasts were the control versus the average of the tween treatments. And then we compared control versus the T80. And then we also compared T80 versus the average of the other two tween treatments to determine whether there's differences related to that fatty acid tail. Looking at dry matter intake, we see that there was a tendency for T80 to decrease dry matter intake by 0.8 kilograms per day, but there were no differences in total fatty acid intake across treatments. As with the first study led by D'Souza, 30 grams per day of T80 increased total fatty acid digestibility compared to control, in this case by 4 percentage points compared to that control treatment. And then T80 in this study also increased total fatty acid digestibility relative to these other two tween treatments. So the fatty acid tail attached does matter. And you can notice that the T40 and T60 treatments had opposing effects here. And recall that T40 is the predominantly palmitic acid and T60 is predominantly stearic acid. So this pattern matches up to that feeding study that D'Souza did, where I showed it in the beginning, where introducing stearic acid had negative effects on total fatty acid digestibility. In this study, we had similar pattern with both 16 carbon and 18 carbon fatty acid digestibility, but I'm not going to show that data. Looking at milk production, the tween had no effect on milk yield. However, all of the tweens increased milk fat yield compared to control, tended to increase milk fat yield compared to control. And that T80 treatment increased milk fat yield by 0.17 kilograms per day compared to control. Conclusions for this third study was that T80 tended to reduce the dry matter intake, but there were no differences in total fatty acid intake. T80 increased fatty acid digestibility and absorption compared to control and to the other two tween treatments. Not all of the tweens improved fatty acid digestibility compared to control because of that tween 60 with the stearic acid tail. So this disagreed with our first hypothesis. However, tween 80 did cause the greatest increase in fatty acid digestibility, which agreed with our second hypothesis. However, we're unsure if this was largely due to the polysorbate or mainly due to that oleic acid effect. So now we've answered the second question, that exogenous emulsifiers can aid the cow in fatty acid digestibility and absorption. Overall summary from my dissertation work, we know that increasing oleic acid reaching the duodenum consistently increases fatty acid digestibility and absorption, and it may improve milk yield or milk fat yield. When we looked at supplying exogenous emulsifiers, we saw that that fatty acid attached to the polysorbate does affect the results. And we also observed that tween 80 increases 16 carbon, 18 carbon, and total fatty acid digestibility and absorption. But again, it's unclear whether this is due to that polysorbate or to the attached oleic acid. Overall, these results will help nutritionists and dairy producers increase absorbed fatty acids and thus energy available to the cow. For next steps, I have both some long-term steps and short-term steps. We did perform another study that actually abomasely infused both oleic acids and tween 80 to examine any additive effects, and I'm still processing those results. And I do have an abstract accepted for ADSA this year for a poster, but I might push that till next year, but those results will be coming out. But in general, I think we need to consider further investigation of emulsifiers. There are many available on the market, both synthetic ones, such as the tween. There's many other synthetic emulsifiers available. There's also the natural emulsifiers, such as lecithin and lysolecithin. And studies have been done with soy lecithin in cows, but none of them have been ruminally protected, and so they've undergone degradation in the rumen. So it's hard to really determine what those effects would be if it was reaching the small intestine. After we examined different combinations and doses of emulsifiers, then I think more product development and testing can begin for an actual product. So I think ideally it will be one product that combines different emulsifiers to have the strongest effect. And this product will likely need to be rumen protected because of those effects seen with lecithin. In the short term, nutritionists and producers can increase oleic acid intake to improve fatty acid absorption. However, they do need to be mindful of the days in milk and milk production body condition score of the cows, as well as the goals of the producer. As with everything, the economics of the marginal return via milk components, health, and reproduction should drive the decision and be continually evaluated and considered for every herd and every group within the herd. I need to acknowledge all of the help on this research from the Locke Lab and dairy farm staff, as well as acknowledge funding from USDA and NIFA and grants from Michigan Alliance for Animal Ag. At this time, I'll take any questions, and I know we are tight on time, so please feel free to email me.
Hey, thank you, Crystal. So while we are switching over to our next speaker, if you release your screen, we'll switch the next one over, but do you mind while we're switching asking, we got one question about, well, what about the effects of the oleic acid supplementation on body condition? Are there any body condition results related to those studies you'd like to comment on?
Yes, so I don't have any. We did look at it in my study, but again, these were just 18-day periods with just 8 cows. So we couldn't make any determinations in the current study. However, other studies, especially work by Jonas D'Souza, has shown that oleic acid does affect energy partitioning. Again, it varies kind of depending on days in milk and energy or milk production of the cow. But oftentimes we do see that oleic acid, the cows will partition some energy towards body condition score, but at the same time, they're often able to maintain the same milk production or pretty close to if they're supplemented with palmitic acid.
Awesome, thanks.